Hi everyone, my name is Dae Kwan Ko. I'm working in the Plant Research Laboratory at Michigan State. I also belong to Great Lakes Bioenergy Research Center. First, I'd like to thank the symposium organizer for inviting me to this wonderful opportunity to talk about my research. Today, the title of my talk is Transcriptional Competition Shapes a Proteotoxic Stress Resolution. Let's talk about that. So, my research mainly focus on gene regulation. The gene regulation is primary response to virtually every biological process. Even though it's a very complex process, the basic principle is very simple, relatively. It's all about interplay between cis regulatory elements and trans elements. Cis elements is called cis regulatory elements called CRE. The short DNA sequence mostly located in the gene promoters. But transacting elements is protein expressed somewhere else and bind to the CRE. Look at this diagram. Gene A has CRE, ATGCGA, and can bound by transvector TF here, and gene A expressed. But gene B has different CRE, ATTTGT, and cannot be bound by this TF, that cannot be expressed. But this is all about interactions between this DNA and the protein. But in reality, gene regulation is not that simple because all these the molecular components, DNA, protein, or RNA, they interact each other, creating biological network. So you can imagine like social networks in our lives. Molecular components, they interact each other to create biological networks. These biological networks allows these complex organisms like plants to coordinate biological response properly. So if genes and CREs and TFs so investigate at a network level, their function could be better understood. So one of the best example is ER stress responses. So let me briefly introduce about ER stress. So ER, so endoplasmic reticulum, is the most important cellular organelle because all the proteins, once they translated, go into the ER and properly folded and dispatch them to the right places. So in normal conditions, just balance between protein folding capacity in ER and protein folding demand. There's no traffic out there. But when plants or other eukaryotic organism express the exposed to stress condition, for example, here drought or flooding or biotic stress or cold heat, the protein folding demands is way increase beyond ER capacity. In this imbalance cause lethal conditions known as ER stress. So ER stress need to be resolved quickly to get back to normal for the organisms. To get that, this organism have special kind of defense mechanism called unfolded protein responses. So unfolded protein response, I'm calling here UPR, is a conserved set of signaling pathway that the purpose is simple to restore ER homeostasis. One thing most important thing is that the actuations and termination UPR are achieved predominantly by gene expression changes in association with many metabolic and cellular events pathway during this ER stress. In plants, there are only two main branches to regulate this complex biological process, UPR. First, it's involved in PG28. PG28 is membrane-associated transcription factor, TF, PG28. And another branch associate the membrane-associated dual functioning protein kinase ribonuclease IL-1 and its splicing target PG60. Once activated PG28 is moving to Golgi, and where it's progressively processed by the cleavages. And the cleavage proteins moving to the nucleus, relocated. And PG60 IL-1 is also activated by misfolded proteins. It splice mRNA encoding PG60 proteins, and then creating the frame shift such that now this splice mRNA can encode these transcription factors, have these nuclear signal targeting. So PG20, PG60 can recognize, can move to the nucleus, and they heterodimerize or homodimerize to regulate their target genes. But growing evidence suggests in reality, the model is not that simple, because mostly PG20, PG60 can regulate each other, also can interact with these other transcriptional regulators. So their transcriptional activities of PG20, PG60 also modulated by interacting with other regulators. So far, only 5 or 6 or few transcriptional regulators found. For understanding of the regulatory mechanism for this coordination of dynamic gene expression by this master regulator, the UPR 2060, 2060, central challenge, because they're partial functional redundancy. 2060, they are partially redundant. Also, the multifunctionality of the TFs in general. Transfer factor mainly have multiple jobs. So even though you remove, the functional redundancy is highly prevalent. Even though you remove one transfer factors, the phenotype is not very much changing. So it's very difficult to identify new regulators in this nature. So to identify new regulators in the UPR, so I took the very unique approaches. First, I look at the gene expression data in ABA treatment, and also ER stress recovery. ER stress recovery means the Arabidopsis seedling was treated with ER stress. ER stress can be induced by specific chemical called tunicamycin. It's very precisely induce ER stress. And we treat them ER stress, and they recover, the transfer this seedling to the media containing just nothing but just gross media, just nutrient. So let them recover. We harvest sample 0, 12, 24 hour time points. We have this RNA-seq profiling, ABA treatment, and ER stress recovery treatment. So there's multiple time points, of course. Then we consolidate all the genes that differentially regulated from multiple time points to a single set. I got thousands of genes differentially regulated in each stress condition. ABA treatment 2600, ER stress recovery 4200. There are a lot of genes differentially regulated. But what's interesting is there's significant overlap between these two data sets. So what I found is there are 928 genes. Those genes are responsible to both ABA treatment and ER stress recovery. And I did go-to analysis. I found out a lot of significant enrichment of the stress and growth metabolic pathway genes. Those genes is very specifically involved in the stress and growth metabolism, very important gene in plants that both respond to ABA treatment, ER stress recovery. We chose ABA for this analysis because ABA is widely associated with these many significant biological processes in plants, stress, growth, metabolism. We found a group of genes responds to both ABA treatment and ER stress recovery. Then I look at their gene expression pattern more carefully. Here is dark line showing x-axis is log to fold changes in ER stress condition, ER stress recovery conditions of 0, 12, and 24-hour time points. And it's log 2 fold change, of course, so over 0 indicating unregulated, and less than 0 indicating the downregulated. And y-axis shows log 2 fold change on the ABA, of course, up and down. What's interesting is so different color indicating different time points of ER stress recovery. On ABA treatment, I have only one time point here. So you can see it's different time points and different color indicating different time points. What's interesting is in 0 hours, there's correlation, positive correlation between ER stress recovery 0 hour data and ABA treatment. What that means is that genes unregulated ER stress condition 0 hours, which means there's no recovery, so only stress condition. They also tend to be unregulated ABA treatment condition. They are downregulated ER stress condition. They also can be likely downregulated ABA stress condition. There's positive correlation. There are significant positive correlation on 0 hours. They look at the 12-hour, 24-hour recovery. The positive correlation is gone. There's no correlation between these two data sets anymore. They're indicating the existence of temporal regulatory link between these and these two stress-induced gene reprogramming pathways, which are potentially regulated by common regulatory factors, which are likely CREs and TFs. So we're interested in now identify CREs and TFs to understand the gene regulatory mechanisms. Then to identify CREs, I did the de novo multiple analysis. The de novo multiple analysis is to grab the DNA sequence, the promoter sequence from the gene, group of genes, 100,000 of genes, and could also grab the genes, the DNA sequence promoters of the randomly selected, same number of randomly selected genes. For example, here we got 928 DEGs responsive to both ABA, ER stress recovery. I also select 928 genes randomly from genomes, from maybe 30,000 genes. Then I grab the promoter sequence of them and then analyze as a control. Then actually in this analysis, I split all the 1 kb promoters with 10 overlapping about 110 base pairs, the fragment. The reason why is that the location matters sometimes. So I got this, that's why I got only 100 base pairs of overlapping fragments and spanning 1 kb promoters. And I named them to 1, 2, 3, 4, all the way to 10, which is close to transfer start site. And what I found was, I found six significantly niched transfer factor binding sites, exclusively niched 3, 4, 5, and 8, 9, 10. These transfer factor sites these potential CREs significantly similar with already known transfer factor binding sites. So this DNA sequence is not random sequence. It's 6 specific sequence could be recognized by transfer factor or transfer factors. Among these, I was really interested. So they're indicating multiple CREs present to specific locations, close transfer start site or far away from transfer start site. They may regulate gene expression in response to both ABA and ER stress treatment. So among them, I was specifically interested in GPAX because GPAX, one of the most interesting, widely known CREs in stress responses, but there's no single report about GPAX in ER stress condition. So I decided to go for the GPAX. Then look at the UPL gene promoters, whether there's GPAX or not. I found multiple copies of GPAX and ABREs. ABRE is core sequence of GPAX, you can say CACG. So ABRE is ABA responses elements. Also, it is core sequence of GPAX. So either GPAX or ABREs, multiple copies, they exist. Either 1 K promoters or 5' UTR. Indicating GPAX is probably the new CRE that we're looking for, modulate gene expression in the UPR. So I was very excited about this result. And then what's next? Then we have CREs that we need to identify transfer factors. So to identify binding target of transfer factors is relatively easy because we can do technical chip, chromatin immunoprecipitation pull down. But the backward, we have DNA sequence. What could be regulated by what could bind this DNA sequence we have is another way. So it's called DNA centered approaches called Easton hybrid. So we trying to perform Easton hybrid. So for this Easton hybrid screening, we choose 3 UPR genes, BG20, BG60. They are master regulator, as I told you before. BG3 is one of UPR gene that are highly induced by ER stress. But BG3 is molecular chaperone genes and the most robust expressions. And the BG3 is known as the representative biomarker genes of the UPR. So BG3 expression inductions means UPR is activated. That's why you choose P3, P28, and P60. So we reason that those genes dynamically regulated under ER stress and others based on our data, published data, and could be probably targeted by multifunctional TFs because those genes are important. And if that, they could link the UPL to other biological pathway. So we wanted to identify transfer factors could be associated other biological process. So through these transfer factors and CRE, we can link a UPL to other biological pathway. So for this Eastern hybrid screening, I got 1 kb promoters, but I also chop up split 2 DNA fragments to overlapping promoter fragments. Then long story short, we got these 6 fragments from the 3 the UPL gene, and we screen these promoter fragments against about 2,000 adapted transfer factors to collect them. Then long story short, we found about a total 603 protein DNA interactions and non-redundant 285 binding TFs from this Eastern hybrid Eastern hybrid screening. So there's a lot of transfer factors we found, and we don't know which one is really binding to the UPL gene promoter in plants. And to do so, I wanted to see kind of network. We have a lot of the interactions, and if you look at this network, you're gonna see where these interactions going, which is proteins, which is genes bound by which transfer factors, so how many bind. So I want to see kind of bird's-eye view. So to do so, I incorporate this protein DNA interaction data with RNA expression data. Previously, I used for this with the ABA's ER stress recovery. So I used ER stress recovery data and then coupled with the protein DNA interactions. So in this network map, you can see each node, the squares, they are transfer factors. And this black circle, they are promoter genes. So PG28, PG60, and this is BIP3. And each square has 3 horizontal heat map strips that indicating gene expression patterns in ER stress recovery, the 0, 12, 24 time points. You can see the blue indicating the downregulated in 0 hours, for example, and this red indicating unregulated. And what's interesting is a lot of genes, a lot of transfer factors, they bind exclusively each promoters. So this protein DNA interaction data is not random. It must be involved in specific DNA sequence. And also here, those transfer factors, all these transfer factors, about maybe 30 to 40, 20, they bound all these genes, 28, 60, BIP3. And these transfer factors bind only PG28 and PG60. And these transfer factors bound BIP3 promoter, but they are bound or P3 or P28, interpret that way. Then I found the high level of regulatory redundancy within UPL genes, and the regulatory module in which multiple transfer factor, they bound either single or multiple UPL gene. The UPL genes regulated by multiple lot of transfer factors. These transfer factors can bind either gene or multiple genes. There are a lot of highly interconnected network. And I look at the biological function of the transfer factor genes. And for example, here, P28 exclusive bound gene, P60 bound gene, P3 exclusive bound gene, transfer factor genes. This is transcription factor GTF genes. They bound all 3 UPL genes. There are a lot of significant biological pathways strongly niche, especially the hormone response and ethylens or a lot of genes. So a lot of metabolic pathways also enrich these genes encoding TFs that bind to UPL genes. So they support our rationale that UPL regulator could be involved in also other stress responses. So we have these bar of transfer factors. Then I choose specific TFs that bind to UPL gene, either P3, P20, P60, or all of them. Also, I choose 12 transfer factor genes based on the association with the gene, binding to the gene, or either or all of them, or association with stress, because I was interested in ABI stress and hormone response based on literature scan. I got these 12 genes, 20 TF genes, and I got homozygous mutant tDNA mutant line, and I checked their phenotype, ER stress responses, recoveries. So one hallmark of ER stress recovery phenotype is that primary root growth. So we treat ER stress for 6 hours and transfer to the growth media without any stress that then grow. Then at the time point, we mark the root tip, then the measure the root growth from that, the tip, the starting point. We also treat the plant at not tunicamycin, which is ER stress inducer. We use the DMS, so it's solvent. DMS is solvent of ER stress induced tunicamycin. So treat the DMS for 6 hours, and then same process as a more control. Then we measure relative growth. While the tunicamycin condition and more control condition, we got the numbers. Then I also another comparison with the Colombia, which is wild type. That's why it's heat map showing. The red indicating just very sick, less growth than Colombia, the wild type, and purple indicating better than Colombia, wild type. So, but here first I use PG20, PG60. PG20, PG60 is already shown to be sicker than Colombia in this ER stress recovery condition. So you can see the red here, then the recover 26, eventually they recover to not like wild type level, but they recover the growth. But they in the beginning they're really sick. This is control. So this experiment worked. Then I look at the other mutant. All those mutant didn't show actually significant difference in this phenotype because probably because functional redundancy of this transfactor is very common. This transfactor they're working as a complex or family genes. Removing one gene may not be suitable enough to make any big changes in the phenotype, except for one mutant, GBF2. So GBF2-3 is one of the knockout mutant of GBF2 gene. And you can see here, this is wild type Colombia, GBF2-3, the mutant. This DMS, so the more control, blue, the red is a clinically treated condition. It grows after 6 days. You can see that the small tip here grows from here like this. Wild type grow about this, this growth, GBF2-3 mutant grow significantly longer than wild type. That indicating this mutant is insensitive to ER stress relative to compared to Colombia, so wild type. Then what is GBF2? So GBF2 is an ABA responsive TFs that binds hundreds of genes via G-box. That's why we're interested to know which temporally regulate gene expression in response to ABA. So GBF2 is key signaling components in ABA signaling pathway, but there's no single report in ER stress. So I was interested about, GBF2 could be involved in ER stress on the UPR gene regulation. So first what I did was I did ChIP-seq, chromatin immune precipitation assay, to identify binding targets genome-wide. The reason why is that in previous study, I already performed ChIP-seq PG20, PG60. The ChIP-seq is a technique that can identify the binding target of potential factor genome-wide. So I already have 2060 data. I wanted to compare GBF2 data. So I did GBF2 data now, and I compared them. Then I got hundreds of genes bound by GBF2. Then I intersect them with PG28 bound gene, PG60 bound gene. Interestingly, the significant overlap between GBF2 bound gene and either PG28, 60 or PG20 and PG60 bound genes. And also I found a G-box is top scoring motif in the GBF2 binding peak, which is demonstrating high quality of my data set. And these highly intersect overlapped gene between 2 data set or 3 data set, they're all involved in, mostly likely involved in the UPR response, ER stress response. So they're indicating GBF2, PG28, and PG60, they co-regulate set of genome-wide UPR gene on the ER stress. It's not about specific couple of genes. Genome-wide genes involved in UPR genes, in ER stress, they co-bound by all these 3 proteins. So to get more mechanistic insight for the functional role of GBF2 in the gene regulation in the UPR, I use PIP3 promoters. The PIP3s, so I mentioned in the previous slide, PIP3 is biomarker genes in UPR. So I look at the PIP3 promoter here. First here, the genome browser is here. The orange is RNA-seq, and this PGP60 chip-seq, P28 chip-seq. It is GBF2 chip-seq data. And also I use DNA's hypersensitive site data as an open chromatin, as proxy of open chromatin. So as expected, in ER stress condition, tunicamycin TM is tunicamycin ER stress inducer, and TM is solvent, more control. In ER stress condition, PIP3 is highly induced, way more induced in tunicamycin conditions in RNA-seq, and also bound by PGP60, PGP28, and GBF to same loci. This location, these peaks containing GFAX. Then also, I didn't bring this here, the data, but I also checked, this all ER stress, ChIP-seq was done, ER stress recovered 0 hours, which means just no recovery, just stress condition. And I also checked GBF2 binding, ER stress recovery conditions, 12 to 24-hour time points. Then I found there's no enrichment, no binding of this GBF to the PIP3 promoters, 12 to 24 time points. And I did gene spatial analysis. Then I found out in GBF2 mutant, in the absence of GBF2, the PIP3 expression significantly, so PIP3 expression induced in Colombia about 7 log to 40 changes. These gene inductions way more significantly enhanced in the absence of GBF2-3. But these gene expression changes was not valid in other time points. And also I used GBF2 overexpression lines. So we use beta estradiol lines, which means beta estradiol can induce GBF2 expressions. I can see here DMSO tunicamycin conditions. But when you look at P3 expression, there's no significant difference between Colombia and overexpression line in DMSO condition. But in tunicamycin conditions, stress condition, when I overexpress GBF2, P3 expression is actually induction was P3 expression. P3 is induced, but induction was attenuated, then indicating GBF2 function as a repressor of P3 expressions in the UPR. So we have this genetic data, we have expression data, we have genome-wide binding data. So we know that they GBF2 bind same location with P26 binding targets mostly. We're talking P3 promoters, but we wanted to know about mechanism. So I hypothesized that GBF2 may compete with P28 or 60 in the binding to P3 promoters because they bind same target. Also P26, they are activator. The GBF2 is a repressor. So I built this hypothesis. So to test my idea, I used P3, GBF2, P28, P60, E. coli purified, MVP, maltose binding protein, the fused the proteins for J-shifter assay. So J-shifter assay, we got protein purified, E. coli purified, also DNA from the fragments. So from the fragments or oligos, we use EW. So EW means endogenous protofragment containing the GFAX. Also I used EM. EM is same protofragment sequence, but GFAX mutated. I use another fragment called EN, which is away from transcriptional site, which just no GFAX there. I use these 3 oligos. First, let's look at this left pictures. First lane 1/1, there's no protein added, only P3 EW, which has GFAX, but there's no protein DNA binding signal, which is expected. Also, I use another control MVP. So in the MVP signal, there's no signal induced, but its typo should be minus. And when I added GBF2, only small amount of GBF protein gets a strong signal of protein DNA in DNA complex. Then indicating even the small amount of GBF2 truly bind to these P3 promoters, which confirm our finding Eastern hybrid and chip-seq data in here, in this case, in vitro. But interestingly, and also when I added EW unlabeled, which means competition assay, not labeled one, so unlabeled one, this binding signal is gradually gone, which means that this binding signal, protein complex signal, is specific to this EW, the oligos. Interestingly, when I added PG28 protein and PG60 protein, the signal is gone. PG60 showed more dominant effects. They're indicating adding 2860 inhibit binding of GBF2, the B3 promoters. And then I had another idea. What about PG60? So when I add, so I here I added PG60 promoters, PG60 protein. I'm sorry. So when I add PG60 protein, the protein signal, protein DNA, the complex signal is increased as expected. And when I added PG28 or GBF2, interest PG28 also has some inhibition effect of PG60, so indicating PG28 they actually collaborating but also inhibiting each other. When I added GBF2 proteins, the binding signal is way weaker than without GBF2 proteins. They're indicating GBF2 compete with PG60 in binding to the B3 promoters. Now we have competition example, and this competition actually matters gene regulation. That's the question I wanted to answer this slide. So test the idea. So we use the transient gene expression analysis using the agro infiltrated the tobacco leaves using the dual luciferase assay system. For this analysis, we got the lipoprotein P3 promoter, 1kb P3 promoter, of course, containing the G box and driving luciferase gene. This is lipoprotein. So we have effectors, 35S promoters, PG60 proteins, splice PG60, PG28 nucleoform, and GBF2. Then I check luciferase activity, whether luciferase activity increase or decrease with this cell. So first lane number one, only P3 promoters, there's no effectors. So this is about a little bit of signal, but this is basal level. When I add the PG60 cell, gets the luciferase activity is increased, significantly increased, indicating P60 activate the P3 promoter, which is expected, as we know. When I add the PG28, the induction levels have way increased. Then P2060, they collaborating to induce P3 expressions, which is already known. But what we didn't know, what happened when we added GBF2. When we added GBF2, there's almost same amount of this cell to either PG20, PG60. There are half of GBF2 compared to the collection of PG20, PG60. There's no significant effect between this number 3 and number 4. When I added double amount of GBF2, which is actually equal to the sum of P20, P60, the cell, there's inhibition effect. So they're indicating not only inhibitory effect of GBF2, the gene regulation B3, but also is showing quantitative inhibitions. So it's quantitative reactions in regulating the gene B3 expressions in this mechanism. So finally, we wanted to see that whether this GBF2 mutation could revert the lethal phenotype PG20, PG60. So PG20, PG60 is lethal in ER stress recovery, which means, when you remove these two master regulator, plant cannot survive, just that, no recover. When I added GBF2-3, another mutation, this double mutant, the triple mutant, the plant partially recovered. You can see that green. So they're indicating GBF2-3 actually suppress the PG20, PG60, suppress the lethal phenotypes. Then what about gene regulation? So check gene regulations, the P3 and ERDJ3B expressions in this triple mutant. Surprisingly, P3 and ERDJ3B, they actually increase. Even though there's no 2860 master regular there, we just remove GBF2, those UPPR biomarker genes, they actually induced. They're indicating those genes also regulated by independent of 2860. All right, so in this presentation, I talked about my recent study. The main question was, how the regulatory dynamics of UPL regulator is achieved. So I took the systems level approach using RNA-seq, histone hybrid chips and C-strom analysis. Then used cis regulatory, cis and trans elements, GPAX and GBF2, for gene regulation the UPL were identified. I also identified mechanism that GBF to compete between PG20 and PG60, which are master regulators the UPR in their binding target, and GBF2 function as a depressor. So our model suggests that a repressive role of GBF2 on UPR gene expressions that monitors the UPR activations. And this study is nice, but bring another question, for example, how GBF2 can sense the right timing, the action to modulate gene expression in the UPR. And also the repressive role of GBF2 and GPAX and the competition mechanism conserved in other multicellular organisms. Addressing this question could enhance our understanding of the UPR gene regulations. So today I talked about many different omics, many different molecular components and using the omics approaches. Recently, I published a series of papers about gene regulation in the UPR, and all these papers simply just collectively suggest highly interconnected gene regulatory networks. I think the rule of thumb seems like keep your friends close, but your enemy closer, because we don't know whether they are actually collaborators or inhibitors. They work antagonize each other. So this must be a top business. So with that, I'd like to thank my postdoc advisors, my Federica Brandis, for her enormous amount of support and personal advice for career path. Also, I was lucky to work with many different research assistants. And currently, I'm working with Lita and Josh. They are undergraduate and also professional research technicians. Histone hybrid screening was done by UC Davis proteomics core with paid service. And this project is supported by mostly NIH grant, also partially DOE grant via a Great Lakes Bioenergy Research Center. With that, I'd like to thank, and please contact me if you have any questions about my presentation. Thank you so much.